I wish to welcome you to this second part of our lecture series on the anatomy and physiology of the urinary system. In this particular session, we are going to look at the structural organization of the nephron, and we'll also talk about the general histologic organization of the kidney. Maybe to bring to perspective where we are in terms of our lectures on the anatomy and physiology of the urinary system. We've already done the first part where we handled the introductory concept regarding the urinary system, and then we narrowed down to the gross anatomical structure of the kidney. The same sitting also handled the functions of the kidney. In this second part, we are going to then look at the histologic organization of the nephron and the kidney as a whole. There's still yet another lecture that will focus on the physiological mechanisms that explain how urine is formed. We call them the three renal processes. The fourth part will be looking at the urinary passages as well as micturition process. And the fifth part will focus on development and congenital malformations of the urinary system. So in this particular one, we're just on the second part. We are focusing on the structural organization of the nephron. We are also going to look at the general histology of the kidneys. Our specific learning outcomes are these one. First, we'll just have a brief review on the basic structure of a section kidney, because that perspective is important for our understanding of the functions of the nephron. We're also going to talk about the three basic renal processes, because these ones influence the structure of the nephron. After that, we will now take longer time to look at the structural organization of the nephron. While we do that, we'll also look at the specific roles of each of the segments of the nephron. We'll also handle the different types of nephrons that we have. We'll see the differences between them. There's something called the juxtaglomerular apparatus. We will look at the components of the juxtaglomerular apparatus and also state the role of the same. We'll then look at the pattern of blood supply through the kidney. This is more of a gross thing, but it's difficult to understand it if you don't understand the full histology of the nephron, and that's why I've chosen to discuss it under this particular section. Lastly, we'll be looking at the microscopic appearance of the kidneys in general. Let's begin with the first one, which is the basic structure of a section kidney. How does a section kidney looks like? So this we did in the previous section, that when you section a kidney, you can describe it in three parts. There's the renal capsule, which is a thin but tough fibrous capsule that protects the kidney. Deep to that is the region we call the renal parenchyma, the region that contains the nephron of the kidney. And then deep to that, we have the pelvic system. The pelvic system is particularly centered within the renal sinus. The renal sinus contains this region with the collecting system, pelvic system, also contain the blood vessels that supply the kidney. We also have some fat tissue as well as nerves. Coming back to the renal parenchyma, which contain the nephron, as we can see in the other image. The renal parenchyma has two zones. The external zone, which we call the renal cortex, and the internal zone, which we call the renal medulla. The renal medulla in particular consists of these conical shaped structures, which we call renal pyramids, with the apex of the pyramid being called the renal papilla, protruding towards the minor calyx, and the base of the pyramid being peripheral. The cortical tissue itself can also be described to have two parts. There's this part of the cortical tissue at the base of the pyramid. That's what we call the cortical arch. There's this part of the cortical tissue that extends between the pyramids. We call this one the renal column. From a histological point of view, the renal cortices are the regions that contain the renal corpuscles, and the renal pyramids lack the renal corpuscles, but they contain the loops of Handle as well as the collecting ducts of the kidney. So basically, that is the structure of a section kidney. Let's remind ourselves then about the three basic renal processes. These are the mechanisms that underlie the formation of urine. So the rate at which different substances are excreted in urine represent the sum of these three processes, whether it's water or minerals or metabolic wastes, whatever is being eliminated through urine represents the sum of these three processes. We call them the mechanisms of urine formation. The first process is what we call glomerular filtration, which is the seepage of plasma from the glomerular tuft of capillaries into the bowmans capsule. These are pressure-dependent events. It relies on some pressure gradients between the glomerular tuft of capillaries and the bowmans capsule. The second one is called the tubular reabsorption. This is the uptake of substances from the glomerular filtrate into the bloodstream. The understanding here is that the filtrate here contains plasma, which is having almost everything, including the ones that you still need. And so tubular reabsorption is an important process of taking back what doesn't need to be eliminated or for other reasons. As long as the substance coming from the renal tubules back to the bloodstream, we call that tubular reabsorption. The third process is what we call tubular secretion. This is the release of substances from the peritubular capillaries back to into the filtrate, and so it's adding to the filtrate. So these are the three mechanisms that explain how urine is formed. Much of the physiology of these three processes is going to be handled in the next topic, which will focus primarily on them. So I've mentioned this here because we will be alluding to them partly when we describe the structure of the nephron and the functions of the different parts of the nephron. And that I thought it's important just for us to have an idea what these processes mean, in as much as the detail of them is going to be handled in the next session. So we can narrow down to the main subject matter, which is to describe the histological structure of the nephron and say the role of the different parts of the segments of the nephron. So when you talk about the nephron, we describe the nephron as the structural and functional units of the kidney. And that is why the region where this nephron is located is actually what we call the renal parenchyma, because the nephron is actually the functional unit of the kidney. Each kidney has many nephrons. We are given about 1 million nephrons, although their number may decrease as we grow older because you cannot regenerate them. Now, even though there are about 1 million nephrons, the number of collecting ducts are fewer. And this is because multiple nephrons drain into a single collecting duct. And in the previous class, we agreed that a collecting duct and all the nephrons that it drains constitute the renal lobule, while one pyramid and the surrounding cortical tissue constitute the renal lobe. So there's a lobe and a lobule. Anyway, when we focus on the nephron itself, each nephron consists of two parts. There's what we call the Rinocopus, which is this region here. And the rest is what we call the renal tubule. So we have Rinocopus and renal tubule. As you can see, the renal tubules drain to the collecting ducts as illustrated in this particular image. All the renal corpuscles are usually located within the renal cortex. However, there are segments of the renal tubules which are found in the cortex, and there are segments of the renal tubules which extend into the medulla, variably though. before it goes back to the cortex. And so that means that the collecting ducts have their portions partly in the cortex before they enter into the medulla. We are going to see which kind of nephrons have their tubules largely within the medulla and which ones do not. The total length of a nephron, if you are to add plus the collecting duct, will range about 45 to 65 millimeters. Now, let's narrow down to the different parts of the nephron. We'll start with the renal corpuscle. The renal corpuscle consists of a tuft of capillaries, which we call the glomerulus, and this surrounding cup-like structure, which we call the bowmans capsule. So these are the two components of a renal corpuscle. The bowmans capsule usually completely surround the glomerulus, actually. As you can see, even in this particular image, the bowmans capsule goes around the glomerulus completely. This corpuscle has two poles. This pole here, where the blood vessels attach, we can see a vessel coming in, we call that the afferent arteriole, and one leaving, we call that the efferent arteriole. So this point of attachment of the blood vessels, afferent and efferent arterioles, is called the vascular pole of the renal corpuscle. And then we have this end of the renal corpuscle, where the renal corpuscle continues with the renal tubule. We call that end the urinary pole. This urinary pole will be marked by a transition of epithelium from squamous epithelium to columnar-cuboidal epithelium there. It's marked by that squamocolumnar junction, as you can see in that particular image. So these are the two poles of a renal corpuscle. Histologically, this is how a renal corpuscle would look like. So you just see a tuft of capillaries and some crescenteric space that goes round like that. The gap within the bowmans capsule is the one we are seeing there, actually. So this end of the renal corpuscle, the one that usually attached with the proximal convoluted tubule, is called the urinary pole. And this other end is where the blood vessel attach. We call that the vascular pole. I want us to discuss the different parts of the renal corpuscle, starting with the glomerulus. So the glomerulus is a tuft of fenestrated capillaries. When you talk about a capillary being fenestrated, it means that the endothelial cells of that capillary have some fenestra, which are holes or pores through the cell. Like in this particular image, remember capillaries usually consist of a layer of endothelial cells and the basement membrane. So if this is the layer of cells and this is the basement membrane, a fenestrated capillary has that basement membrane very intact, as we can see in that image. Also, we have a layer of endothelial cells which are attached to one another. However, we have holes through the endothelial cells. These holes are the ones we call fenestra, and such a capillary is termed fenestrated capillary. Maybe just to help you understand the histological types of capillaries. So the middle image shows us a cross-sectional outline of a fenestrated capillary. This one being the basal lamina, which is continuous. And then this one shows us the endothelial cell. So the breaks you're seeing there are the gaps between within the holes through the endothelial cells, the fenestra. So this is the fenestrated capillary. Compare that with this kind of capillary, which we call continuous capillary. The basal lamina is continuous, as you can see, and the endothelial cell is also continuous, doesn't have any break within it. Also compared it with sinusoidal capillary, which is also called discontinuous capillary. We can see breaks in the basement membrane, as well as breaks within the endothelium itself. So in the glomerulus, we have fenestrated type of capillaries. The other regions where we have fenestrated capillaries are usually the villi of the small intestine, as well as also in endocrine glands. Anyway, that is just to help us understand what a fenestrated capillary looks like. The fenestra within fenestrated capillary, the fenestra, the windows, the fenestra within fenestrated capillaries make fenestrated capillaries like the ones in the glomerulus be exceptionally porous. And this one help to allow large amounts of plasma to pass through. And we know that virtually almost everything in the plasma can pass through apart from the plasma proteins. The reason the plasma proteins do not pass through is because they're large and also negatively charged. There's something we're going to see that make negative charges not go through. So anyway, the fenestra allow large amounts of fluid to pass through the process of glomerular filtration. So anyway, that is the glomerulus. Apart from the glomerulus, the other component of the Rinocopus is the bowman's capsule. The bowman's capsule has two layers. There's an outer layer here, which we call the apparatus layer, and the inner layer, which we call the visceral layer. The inner layer cling closely on the walls of each individual capillary tuft. And that's why it's seeing the outline. It's following the outline of the capillaries. But the outer layer is that one, the apparatus layer. The inner layer is called the visceral layer. The gap between them, the space between them, is what we call the bowman's space or the urinary space. Histologically, the apparatus layer of the bowman's capsule consists of simple squamous epithelial lining, as you can see there, one layer of flat cells. This simple squamous epithelium is continuous with the proximal convoluted tubule at the urinary pole, and that is where we have the squamocolumnar junction. The visceral layer of the bowman's capsule clings on the glomerular tuft of capillaries. Usually, the cells that form the visceral layer of the bowman's capsule are highly specialized. They are highly branching cells, which we call podocytes. These podocytes cling closely and intimately with the basement membrane of the capillary endothelium. This image captures for us that aspect. So this is the the vascular pole with the afferent and efferent arteriole. This is the urinary pole. Squamocolumnar junction is demonstrated there. So this is the apparatus layer of the bowman's capsule, simple squamous. Now the visceral layer, we can see these colored cells enveloping around the tuft of capillaries. These specialized cells are the ones we call podocytes. These podocytes have several processes, primary, secondary, and tertiary processes. These processes are the ones which go around the basement membrane of the capillaries. In the manner shown in this particular image, we can see perhaps this is a primary process, and maybe that's a secondary process, and that's a tertiary process of the podocytes. So we can see the process of the podocytes go around the capillary. And in this image, we can also appreciate that they interdigitate. These processes interdigitate with one another, leaving some gaps between them. The gaps, the clefts between the processes of podocytes are known as filtration slits. It is through these gaps that actually fluid enters the bowman's space. And so I want you to understand that really well, that we have fluid running within here, that is plasma, blood. And so blood will go, the plasma will go through the fenestra of the capillaries, then go through the basement membrane there, then go through the filtration slits of the podocytes. This image captures for you the podocytes as they envelop around particular fenestrated capillary of the glomerulus. Great. Having said so, now let's discuss the filtration membrane. We can call it the glomerular filtration barrier or the filtration membrane. It is the tissue interface that lies between blood and the interior of the bowman's capsule. It is the one where the structures through which plasma will pass as it is being filtered from the bloodstream into the bowman's space. So the filtration barrier consists of, if we remember, we mentioned that blood will be flowing through here. It will have to go through this cell, then the basement membrane, then the podocyte processes. So we can say that the filtration membrane consists of the following. One, the fenestrated capillaries of the the fenestrated endothelium of the glomerular capillary. Simply, it means that the endothelium of the glomerular capillary is one of the components of this barrier. But remember, this endothelium is fenestrated. So the plasma will go through the fenestra, actually. The plasma goes through the fenestra. After the plasma has gone through the fenestra of the endothelium of glomerular capillaries, it will pass through the basement membrane. The basement membrane in the glomerulus is a fused basal lamina formed by the basal lamina of the endothelium of the capillaries and the basal lamina of the podocytes, because podocytes are also epithelial cells. So they also have a basal lamina. The fused basal lamina is what we are calling the glomerular basement membrane. This fused basal lamina is actually a physical barrier. This physical barrier is usually negatively charged. And these negative charges that it contains, because of some chemicals that it has, make the glomerular basement membrane be repulsive to substances which are negatively charged. And so for that reason, proteins are still further prevented from passing through this barrier, because proteins are examples of anions. They are large and also negatively charged. So that's another reason why proteins are kept away from filtration. Anyway, the third component of the barrier are the food processes of the podocytes. Remember, the food processes of the podocytes have gaps between them. Usually, these gaps are filled with some thin membranes. Those thin membranes are called slit diaphragms. And so the slit diaphragms also help to filter substances which could have passed through the fenestra of the endothelium. If the man to pass through the fenestra of the endothelium, then also pass through the glomerular basement membrane, they can still be further trapped by the slit diaphragm of the podocytes. So these three are the structures that constitute the glomerular filtration barrier. Look at it this way from this image. So consider this to be the inside of the capillary, with the capillary endothelium being this one here. So these are the gaps, the fenestra, that one. So once the filtrate moves from the bloodstream, plasma moves through there. It will pass through this region. This region is the fused basal lamina, formed by the basal lamina of the endothelial cells and the basal lamina of the podocytes. And then after going through the first basal lamina or the glomerular basement membrane, it will go through the filtration slits that are found between the podocyte processes. So these three structures constitute the barrier. Remember, the filtration slits have some thin membranes, which we call the slit diaphragm. And so large substances which have passed through here and here may still be trapped at this level. All in all, that is the filtration membrane. Those three structures constitute the glomerular filtration membrane. This danger of the filtration membrane being clogged by substances as time goes by. And for that reason, the filtration membrane is usually cleaned by some phagocytic cells within the glomerular tuft of capillaries. We call them the mesangial cells. Mesangial cells are the phagocytic cells within the kidney. Apart from cleaning the glomerular filtration membrane, they also have other functions as well. This image captures for us a glomerulus, the Rinocopus with the urinary pole and the vascular pole with the two vessels, afferent and efferent arterial. Within here, we see tuft of capillaries, but we also note that we have some mesangial cells there which help to clean the barrier. Great, so that's the filtration barrier. And that summarizes for us the story about the Rinocopus. We've looked at the glomerulus, we looked at the bowmans capsule, and we looked at the filtration membrane. Now let's talk about the renal tubules. This is the other part of the nephron. The renal tubules, a single renal tubule will be about 3 centimeters long. That is really long. And has 3 major parts: the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. So those are the 3 major parts of a renal tubule: the PCT, the loop, you can call the loop of the nephron or the nephron loop, or the loop of Henle, and the distal convoluted tubule. In this image, this is the proximal convoluted tubule. This is the loop of Henle, and that's the distal convoluted tubule. So this distance from there up to there is about 3 centimeters. You'll remember that the whole thing can be even up to 6 centimeters. Now, throughout its length, the tubule, together the collecting ducts, are just made up of a single layer of epithelial cells that lie on a basement membrane. So the whole of the tubule, just one layer of epithelial cells. And that is why we are saying that the parenchyma consists of the epithelia of the nephron, just a single layer of epithelial cells. Whether we're talking about the renal tubule or we're talking about the collecting duct. Each region, however, has some unique histological characteristics that will reflect the role of that particular region in terms of how that region processes the glomerular filtrate. And so for that reason, I want us to narrow down to each of the regions of the renal tubule and see what's unique about them. Let's start with the proximal convoluted tubule. Yes, it is true that the proximal convoluted tubule has one layer of epithelial cells, but what is the structure of these epithelial cells? They're largely cuboidal cells or local lamina cells. These cells have a lot of mitochondria within them, reflecting how active they are metabolically. And they have numerous elongated microvilli, which makes the cells of the proximal convoluted tubule have a very definite, thick brush border. This image captures for us an example of cells that are found within the proximal convoluted tubule. we can see the dense microvilli on the apical zone, usually very long. We can see the numerous mitochondria within the epithelium, and we can see basal membrane invaginations as well, representing active absorption. So usually you'll call this the apical zone or the luminal zone because the renotubule lumen is on this side, and we call this on the basolateral side. So the basolateral side of the epithelium has several infoldings still as an adaptation for active transport of substances. Anyway, so that's how the cells with the PCT look like. Remember, the PCT is highly convoluted as well to basically just slow down substances that pass through. From there, we have the loop of Henle. The loop of Henle has thin segments as well as thick segments. The thin segments are lined by squamous epithelium, and the thick segments are lined by low columnar or cuboidal cells. And basically, the thick segments reflect or look similar to the structure of the adjacent convoluted tubules. So the thick segment next to the proximal convoluted tubule is a thick descending segment. The structure is the same. The thick segment that is ascending near the DCT also has a similar structure like that of the DCT. And so it will also almost appear like that one. It will be largely almost like PCT, except that the cells are shorter and they don't have microvilli, or if they do, they're very sparse. So this is the structure of the DCT, as well as the structure of the thick segments of the loop of Henle, the thick segment that is adjacent to the DCT. Then the thick segment that's adjacent to the PCT will be similar to the structure of the PCT. The thin segments of the loop of Henle just made up of squamous epithelium. That's the structure of the renal tubule. So this image helps us to understand all of them in one image. If this is the PCT, we can see the cells there have several microvilli, a lot of mitochondria, and basal membrane invagination. That is PCT. And that will also extend up to this segment here, the segment of the loop that is going down, but is still thick. Then we have the thin segment. This thin segment of the loop has squamous epithelium. Then this thick segment of the loop will have almost similar structure like that of the DCT. And so basically lower cells with fewer microvilli, if any, but they still show a lot of mitochondria and basal membrane invaginations. And then moving from there, we can now talk about the collecting ducts in the next slide. Maybe just something to mention about the DCT. We can see that the DCT makes contact with the Rinocopus. Look at the way the loop of Henle goes back to the parent Rinocopus. So at the termination of the loop, at the beginning of the DCT, there's a region there which contains some specialized cells, which we call macular denser cells. These macular denser cells are chemoreceptor cells of the DCT. Very important in some regulatory functions of the kidney, as we're going to see. Now let's talk about the collecting tubules and the collecting ducts. We generally call this part here, almost entering the collecting duct, the collecting tubule is the straightened part of the DCT. So that collecting tubule, or some people call it connecting tubule, and the collecting duct, I'll discuss them together. The collecting duct itself is about 2 centimeters long and receive multiple nephrons. the collecting duct will pass through the renal cortex first, then pass through the renal medulla. Remember, the parts of the collecting ducts that are extending into the cortex are the ones that constitute what we call the medullary rays. Then the parts that extend into the medulla, multiple of them will join together. The larger ones join together to form what we call papillary ducts. So the papillary ducts are formed by union of collecting ducts as they go towards the renal papilla. As the collecting ducts pass through the medulla, the pyramid, it will give the pyramid some striated appearance. Each collecting duct still has epithelial cells on its wall, but there are two types of cells within the wall of collecting duct. We have what we call the principal cells. You can call them P cells. These are the majority of the cells. They're usually relatively taller, but they have fewer organelles. These are the cells which contain the receptors for antidiuretic hormone. They are particularly involved in reabsorption of sodium as well as water, but the water reabsorption is dependent on the presence of antidiuretic hormone. So we can say they're involved in fluid and electrolyte hemostasis. These are the principal cells. So there are very few microvilli, if any, but they're usually a bit taller. There are few organelles as well. Compare that with the second cell type there, which we call the intercalated cells. You can call them eye cells. The eye cells have numerous microvilli, as you can see there. They have a lot of organelles with a lot of cytoplasmic vesicles. These particular cells are the ones involved in acid-base homeostasis. So they basically secrete acid, and they're also important in transportation of bicarbonate reabsorption, basically. So remember, the principal cells are involved in fluid and electrolyte homeostasis, while the intercalated cells are involved in acid-base homeostasis. Great, so we've described the nephron, but let's just make a summary of what we've said about the structure of the nephron. We've mentioned that the nephron has a region called the Rinocopus, which consists of the glomerulus with an outer layer, apparatus layer, and inner layer, visceral layer, although the visceral layer envelop each capillary. And the capillary that enters is what we call the glomerulus, with this being the vascular pole and this being the urinary pole. The vessel entering is called afferent arterial, the one leaving is called efferent arterial. This is the Rinocopus. The Rinocopus is the main site of plasma ultrafiltration. And so we talked about the structural adaptation that the Rinocopus has that enable to do this. The presence of fenestrated capillaries, the presence of podocytes with filtration slits, and also the fact that this barrier is usually cleaned by the mesangial cells. The filtrate enters the renal tubules, and the first segment of the renal tubule that the filtrate enters is called the proximal convoluted tubule. That's the general outline. It is convoluted to slow down substances as they move along there. Structurally, the cells here have a lot of microvilli to promote absorption. They also have a lot of mitochondria, which help in active transport of various substances. The PCT is the principal site of tubular reabsorption. About 65% of tubular reabsorption occurs here and can even go to 100% for some substances. From there, we have the loop of Henle. So the loop of Henle has a descending segment and an ascending segment. or we can say the descending limb and the ascending limb. And each of the limbs have thick and thin segments, but I'll say something about that shortly. So the thick segment that's going down, you can call the thick descending limb of the loop of Henle. The structure of this one is similar to that from the PCT, and perhaps also similar functions. And then we have the thin descending limb of the loop of Henle, which is lined by squamous epithelium. And then this part here, the thin ascending limb of the loop of Henle. And then this one, the thick ascending limb of the loop of Henle. Some nephrons don't have this particular segment here. So the thick ascending just begin from down here, and I'll tell you which type of nephrons have that kind of structure. The structure of the thick ascending limb is similar to the structure of the DCT. So those are the segments of the loop of Henle. Generally, based on the various functions of the loop, and we're going to see that in the next topic, there are some variable transport mechanisms that occur around the loop that are aimed towards concentration of urine. So the structure of the loop of Henle is geared towards urine concentration. That's perhaps the major function of the loop of Henle, participate in the urine concentration mechanisms of the kidney. From the loop, we have the distal convoluted tubule. The distal convoluted tubule, again, is convoluted. The structure is similar to this of the PCT, except that it doesn't have microvilli or hardly any. Now, the junction between the thick ascending limb and the DCT makes contact almost with the Rinocopus, the parent Rinocopus, and that region contains some specialized cells which we call the macular denser cells. The key role of the macular denser cells is that they are chemoreceptor cells. Their major role is to detect the concentration of sodium within the renal tubules. And this is important in providing information for that particular nephron that help it to guard or to control the amount of blood flow that enters through the glomerulus. And that in turn will control renal blood flow as well as controlling the glomerular filtration rate. So this information that relies on the concentration of sodium within the renal tubules to control the blood flow to the kidney is basically what we call the tubular glomerular feedback mechanism. It's a feedback mechanism that helps to regulate the renal blood flow as well as glomerular filtration rate. We will talk about it again shortly in a deeper way. Well, not shortly, but in the next one, in the next topic, we'll talk about it. We can say that that's a key function of the DCT. Well, apart from that, the other segments of the DCT do not really have the macular denser cells. So these are the segments of the DCT which are not really contain the macular denser cells are still important in tubular reabsorption of some ions, and especially hormonally regulated for that matter. Then we have the connecting tubule and the collecting duct there. The collecting tubule and the collecting ducts simply collect urine, and remember they'll merge and form the papillary ducts which empty into the minor calyces. Remember the cells of the collecting ducts, two types. The P cells are largely for homeostasis of water and ions, and that will help in urine concentration. Remember, urine concentration is a mechanism of water conservation. The I cells, on the other hand, are responsible for acid-base homeostasis. So that's the structure of the nephron. We talked about that as well. And so I think you put that into perspective. Remember the epithelial type of the collecting duct. still simple columnar, simple cuboid. Usually, the collecting ducts near the cortex will be more cuboidal, and the ones deeper towards the papilla will be more columnar. Now we can talk about the different types of nephron. There are two types of nephron. We have what we call cortical nephrons and juxtamedullary nephrons. The cortical nephrons have their renocorpuscles in the outer zone of the renal cortex, as you can see in this particular image. Usually their tubules are short, as we can see here, and the tubule is predominantly within the renal cortex. Just a small part of it may loop into the medulla, but most of it is in the renal cortex. Importantly, also notice that their tubule lack the thin ascending segment. So the ascending segment of the loop is largely the thick ascending segment. It doesn't have the thin ascending segment. These are the majority of the nephrons, really, which are responsible in the excretion of substances. But we have the juxtamedullary nephron, which is now the other one there. Although the renocorpuscles are in the cortex, they're in the inner zone of the cortex adjacent to the medulla. And that's why we are saying they're juxtamedullary. The renocorpuscles are near the medulla, not within, but near the medulla. Basically at the corticomedullary junction. The loop of Henle, or rather the tubule, including the loop of Henle, is really long for this particular nephrons. And look, the loop is largely within the medulla. Very long loops of Henle and largely within the medulla. Also important to note is that it has a very prominent thin ascending limb of the loop of Henle. This kind of nephrons constitute about 15% of all the nephrons, and they're the ones which are vital in the urine concentrating mechanisms of the kidney, as we're going to see in the next topic. Great, so these are the different types of nephrons. Now let's talk about the juxtaglomerular apparatus. What are juxtaglomerular apparatus? Juxtaglomerular apparatus are located where the renal tubule makes contact with the parent renal corpuscle, as I mentioned earlier, that the renal tubule usually makes contact with the parent renal corpuscle. In particular, it will be the junction between the thick ascending limb and the DCT. That junction between the termination, at the termination of the thick ascending limb and the beginning of the DCT, that is the region that makes contact with the parent renal corpuscle. And so that junction is important in some mechanisms of the kidney, which we call the tubular glomerular feedback mechanism. This feedback mechanism is important in maintaining an inverse relationship between the concentration of salt within the renal tubules and the glomerular filtration rate. What that statement simply means is this, that when situations come that the amount of salt being delivered within the DCT is low, the feedback mechanism will work to increase the GFR. But if the amount of salt load being delivered is very high, the feedback mechanism will operate to lower the GFR. So there's an inverse relationship between salt concentration and the GFR. This is the feedback that maintains that inverse relationship. And it is through this that the kidney is able to actually monitor the amount of salt being delivered It operates in maintaining a constant amount of salt being delivered. And through that is able to regulate blood pressure into the kidney. It's also able to regulate the glomerular filtration rate. What are the cellular components of the juxtaglomerular apparatus? We have the macular denser cells that I mentioned earlier. They are chemoreceptor cells. They are just specialized epithelial cells, but they're chemoreceptor cells. Their role is to detect the concentration of sodium chloride within the renal tubule. Now, the point is this, that this region reabsorbs sodium chloride. So if amount of sodium chloride passing through it is a lot, then it will know that it's actually a lot in the tubule. And if it's little, then it will know that it's actually little in the tubule. So based on the amount of sodium chloride being reabsorbed, the macular denser cells are able to know whether we have high or low sodium within the renal tubules. The second component of the apparatus are the ones we call the granular cells, or you can just call them the juxtaglomerular cells. That's the commonest name. These are specialized smooth muscle cells found within the wall of the afferent arteriole mainly, although a few may also be found in the wall of the efferent arteriole, but mainly in the wall of the afferent arteriole. These cells are responsible for secretion of renin enzyme. Renin enzyme, as we saw in the previous talk, is responsible for converting angiotensinogen to angiotensin 1. Then in the lungs, angiotensin 1 will be converted to angiotensin 2. The third component of the apparatus is what we call the lysis cells, or you can call them the extraglomerular mesangial cells. So these are mesangial cells outside the glomerulus. That's why we call them extraglomerular mesangial cells. We call them lysis cells because they form like a lace network between the two cells. That's why it's called the lysis network, the lysis cells. Now, the role of the lysis cells is to basically form a path through which we can have signal transmission from the macula densa to the juxtaglomerular cells. So it forms a path where the macula densa can actually communicate with the juxtaglomerular cell in terms of perhaps stimulating renin secretion or inhibiting renin secretion, depending on the concentration of sodium here. So for example, if the amount of sodium within here is low, then macula densa cells will perhaps send a signal for secretion of renin. But if the amount of sodium here is high, it will inhibit renin secretion. Great, so these are the components of the juxtaglomerular apparatus. We'd already talked about the renin, angiotensin, aldosterone system in the first talk, but there's some components of this, like the tubular-glomerular feedback, that we're going to talk about better after we describe the glomerular filtration in the next class. Now let's talk about the pattern of blood flow through the kidneys. The kidneys receive the renal arteries, as we mentioned in the first class. A lot of blood actually flows through the kidney. If you are to look at the amount of blood that goes through the kidney compared to the cardiac output, we see that the kidneys receive over 22% of cardiac output. That is even higher than what the brain actually receives. Well, the high amount of blood that flows through the kidney is not just for metabolic need of the kidney, it's largely for the other function of the kidney of excretion of substances and controlling fluid volume. Most of the blood that goes through the kidney actually is directed towards the renal cortex as opposed to the renal medulla. And in the next class, we are going to see why the renal medulla does not need a lot of blood supply. So the amount of blood that goes through the renal medulla is less than 5% actually. Most of the blood flow goes to the renal cortex, where we have the rinocopus. So how does the renal artery divide when it enters? The renal artery is a branch of the descending aorta, the abdominal aorta. So we have a right and a left renal artery, each going to the particular kidney of that side. So the renal artery at the hilum usually divides into two. We have an anterior division and a posterior division. The anterior division divides further into four, and those four are called the segmental arteries. The posterior division usually does not divide into further segmental artery, and so the posterior division continues as the posterior segmental artery. But the anterior division gives us four segmental arteries. And so we have 5 vascular segments of the kidney, 4 in front and 1 behind. These are the names given to the vascular segments of the kidney. The ones from the anterior division, there'll be a segment that is going to the upper pole of the kidney, so that the superior vascular segment. There'll be another one that goes to the inferior pole of the kidney, that's the inferior vascular segment. And then there'll be 2 that will be going in the anterior, directly anterior, one on the upper part, so the anterior superior and the anterior inferior segment. So these 4 are segments from the anterior division. Then the one from the posterior division is just a continuation, it's only 1. So there are 5 vascular segments of the kidney. The segmental arteries then divide, as we can follow from here. Let me change that pointer so that you can see better. Okay, so if this is the renal artery, and so the divisions and the segments, let's take this to be a segmental artery then. This segmental artery now give you arteries that run between the pyramids. Remember, a renal pyramid and the surrounding cortical tissue constitute the renal lobe. So the segmental arteries give you arteries that run between the lobes. Therefore, we call them the interlobar arteries. Segmental arteries give you interlobar arteries, like this is an interlobar artery. The interlobar arteries run between the pyramids, between the lobes. At the junction between the base of the pyramid and the renal cortex, the interlobar arteries give you some arteries which are given at around 90 degrees from the artery. We call them the arcuate arteries. Arcuate arteries usually are considered end arteries. This is because we know that we don't have communication between one arcuate artery and another one. There are no anastomosis between the arcuate arteries, and so arcuate arteries are end arteries. That means if you knock off one, that region supplied by that arcuate artery actually can undergo ischemia. Well, from the arcuate arteries, we have some arteries that run from that junction, corticomedullary junction, towards the renal capsule. We call those ones cortical radiate arteries because they radiate towards the renal, through the renal cortex towards the capsule. The cortical radiate arteries are running between the lobules, and so you can call them interlobular arteries. So the cortical radiate arteries like these ones, or those tiny ones, are the ones we are calling the interlobular arteries. Now look at this again. So if this is the interlobar artery, it will give you the arcuate artery that run between the corticomedullary junction. The arcuate artery then gives you the cortical radiate artery, which we've called the interlobular artery. The interlobular artery is the one that gives you the afferent arteriole that go to the renal corpuscles, as we can see in these particular images. So the afferent arteriole goes to the glomerulus, and from the glomerulus you have efferent arteriole. Maybe this image would be better to capture that. So this is the afferent arteriole which enters the glomerulus, and then from the glomerulus we have efferent arteriole. The efferent arteriole then gives you what you call peritubular capillaries. peritubular capillaries are capillaries that go around the renal tubule. However, there's a special type of peritubular capillary that follow the length of the loop of Henle along the juxtamedullary nephrons. That type of capillary that follow the length of the loop of Henle within the renal medulla is what we call the vasa recta. So yes, you have peritubular capillaries that go around the renal tubules, but the capillaries that specifically follow the length of the loop of Henle as it enters the renal medulla, and this especially applies for the juxtamedullary nephrons, this is called the vasa recta. Look at the direction of blood flow within the vasa recta, compare that with the direction of urine flow. You'll see that is actually flowing the opposite direction. Very important to note. Anyway, so we have the vasa recta, which follow the loop of Henle. They are still capillaries anyway. So from those capillary network, the veins join again, reconstitute eventually in the same reverse pattern until we have the renal vein, which enters into the inferior vena cava. So this image summarizes that story from the outer renal artery. Remember, there was an anterior and posterior division here, then segmental, there are 5 segments, then interlobar, then aquatotary, then corticoradiate, which is also called the interlobular, which gives the afferent arterial, then glomerulus, then efferent arterial. Then we have the peritubular capillaries of the vasa recta, and then going again, forming the veins back to the renal vein, then drain to the inferior vena cava. What do we notice? That there are 2 capillary beds within the kidney. The first one is at the glomerulus, and the second one is in the peritubular capillaries, connected by the efferent arterial. Remember, the presence of 2 capillary beds within one vascular circuit is what we call a portal system. And so the kidney is also another site where we have a portal system. Remember the other portal systems in the body. We have the one between hypothalamus and the pituitary. We call that hypothalamo-pituitary portal system, the one that is responsible for transportation of releasing and inhibiting factors from the hypothalamus to the anterior pituitary. The most elaborate of the portal system is the hepatic portal system, the vascular connection between the capillaries within the wall of the GIT and that of the spleen with the capillaries within the liver, joined by the hepatic portal vein. All right, so this is the renal blood flow. Maybe let's just say something about this renal blood flow. There are some factors that determine the amount of blood that flows through the kidney. One would be the blood pressure within the renal artery. And the blood pressure within the renal artery will definitely be influenced by the systemic blood pressure. So if the systemic blood pressure is high, the arterial pressure within the renal artery is going to be high. If the systemic blood pressure is low, even the pressure within the renal artery is going to be low. And that will affect perfusion of the kidney, although within some physiological range. So within extremes of physiological range, that is. So this is what I mean. If the pressure is too low, the renal blood flow will be low. If the pressure is too high, the renal blood flow will also be high. But there's a physiological range within which changes in systemic blood pressure is not going to affect the renal blood flow. We call that renal autoregulation. We'll talk about it in the next session. The other parameter that affects renal blood flow is the pressure within the renal vein. It is usually about 3 to 4 millimeters of mercury, but in pathological conditions where there is perhaps some blockage or obstruction, for example, the pressure within the renal vein can be high. And if the pressure within the renal vein is high, maybe because there's a clot or a tumor there, then you're going to affect renal blood flow. Because high pressure within the renal vein will lower renal blood flow, and low pressure within the renal vein will increase renal blood flow. Remember, blood flow is usually the pressure gradient between 2 points divided by the resistance, which is the third parameter actually that affects renal blood flow, the vascular resistance through the kidney. And this will be determined by the various vessels, especially the interlobular, the afferent, and the efferent arterioles. Those are the key determinants of total vascular resistance within the kidney. So though the resistance offered by those vessels will also affect blood flow. If the resistance is high, the flow is low. And if the resistance is low, the flow is high. Remember, flow F is equals to pressure gradient between 2 points divided by the resistance between those 2 points. So I've told you that although changes in the arterial pressure influence renal blood flow, the kidney has some mechanisms that help us to maintain renal blood flow, and that will also maintain the glomerular filtration rate at a constant rate within some range of pressure. And the range of pressure here is a mean arterial pressure of between 80 to 180 millimeters of mercury. If the mean arterial pressure within that physiological range, then the kidney is able to adjust the renal blood flow to a relatively constant state. That means that changes in systemic pressure, does not affect much of the renal blood flow as long as the systemic pressure is within that particular range. We call that renal autoregulation, and there's some mechanisms that help in renal autoregulation that we'll discuss in the next topic. Now, maybe just take a minute to look at the question projected to you. Just try to answer some of these things. You have some things labeled there from 1 to 24. Take a minute, see if you can be able to name them and just identify them. For you to know them, you'll need the knowledge of what we discussed in this session, as well as what we discussed in the first session. Okay, I hope that you've oriented yourself on that. Now let's look at the last part of this lecture, where we just make a summary of how the kidney looks like histologically. So the slide projected to you here, the slide of kidney, although not from human. And that is why you are seeing that it has only one pyramid. So it's not from human, but the histology will still be the same. So the kidney consists of, when you cut across the slide of the kidney, what makes you know that the slide of the kidney? A number of things. One of the most obvious things that we usually see is you see several tubules cut in cross-section. And perhaps that's the most marked structure thing that you see on a slide of the kidney to help you know that this is the slide of a kidney. Tubular structures, multiple tubular structures cut in cross-section. And those tubular structures are just made up of simple epithelium. Now, let's put into perspective the different parts of the kidney that we talked about and how they look like histologically. We start with the renal capsule. The renal capsule is a connective tissue structure. So it's a dense, regular connective tissue structure that cover, therefore protect the kidney, and the one labeled D there, that's the renal capsule. So we can see that this highly tubular structure is covered by a connective tissue structure. So that's the renal capsule. Even here, that's the renal capsule. Deep to the renal capsule, now we concern with the renal parenchyma, which we are going to talk about more. So this is the renal parenchyma, really. Remember, the outer zone of the parenchyma is what we call the renal cortex. We would expect the renal corpuscles to be in this region, and the inner zone is called the renal medulla of the pyramid. We don't expect the corpuscles here. Towards the renal sinus, we have that space with the calyceal system. In this case, because it's just a single pyramid, so that would be the renal pelvis, actually. Maybe we magnify a bit, and so you're able to see that the outer region, the renal cortex, actually contains some structures, which we call the renal corpuscles. You'll see them better when you focus on the cortex. The medulla doesn't have the corpuscles. Now let's begin with the renal cortex. So the renal cortex is the outer zone of the parenchyma. It contains a number of things. One, the renal corpuscles, as you can see them, numerous. A higher magnification of the renal cortex shows you the corpuscles as well. Apart from the renal corpuscles, the other things which are contained within the renal cortex is this region here, which we call the cortical labyrinth. Remember, the cortical labyrinth consists of the proximal and the distal convoluted tubules of the kidney. So these regions are the renal corpuscles, but between the corpuscles, we have the cortical labyrinth, consisting of the DCT and the PCT. And those ones are separated by some striations, as you can see here. These striations are elongated tubules, which extend from the renal medulla into the renal cortex. We call them the medullary rays. The medullary rays represent the proximal segments of the collecting ducts. So the cortical segments of the collecting ducts are called the medullary rays. So the renal parenchyma, if you have to look at the part of the nephron that are located within the renal cortex, there are three: the renal corpuscles, the cortical labyrinth, and the medullary rays. I'm not mentioning the blood vessels, but of course you know that they're also there. I'm focusing on the parenchymal parts. This is a high magnification of the same. So that's the renal corpuscle. This is the cortical labyrinth, and this is the medullary rays. If you're to narrow down to a single renal corpuscle, we see that this is the tuft of capillaries, and this is the bowmans space. Look at the outer layer of the bowmans capsule. You'll appreciate that it's lined by simple squamous epithelium. And then we have the bowmans space. Remember, the podocytes are within this tuft, together with mesangial cells and capillary endothelium as well. This is a high magnification of the renal corpuscle to just help us understand the epithelial lining of the parietal layer of the bowmans capsule, simple squamous epithelium. This is showing us the region of the cortical labyrinth with the intention of us trying to pick the difference between the PCT and the DCT. Now, when you look at it, you might think it's difficult to know what is PCT and DCT, but it's actually possible. First, understand that if you're picking it from the cortical labyrinth, it means you only have a DCT and a PCT. We don't have the collecting duct here. So that's the first point to understand. So with that understanding, we only have PCT and DCT in this zone. Now, what was the main histological difference between a PCT and a DCT? The main histological difference was based on the fact that the PCT, the cells of the PCT are taller and have numerous microvilli. The cells of the DCT are slightly shorter and have hardly any microvilli. So we are using that concept to distinguish them. Therefore, the ones which are PCT will look like the lumen is slightly smaller and there are extensions into the lumen. They have a brush border. And then the ones which are DCT will have a well-circumscribed wall without those extensions. So here, this is most likely a PCT, and this is most likely a DCT. Or this is most likely a DCT, or this is most likely a PCT. Maybe let's use another one. So this one shows better, much better. So here, that's a Rinocopus anyway. And so there's the cortical labyrinth. So this region is being magnified for us on this other end. We're able to see. Now look at the DCT. The walls are very clean with wider lumen, but the PCT like that, or like that, or like that, the walls are, the lumen are smaller and there are extensions into the lumen. So that's how we distinguish PCT and the DCT. Now we can now look at the renal medulla, and in particular we are focusing on a renal pyramid. So this is a renal pyramid. Appreciate that we do not have Rinocopus here. Also appreciate the presence of vessels between the cortex and the medulla. So these vessels here running at the corticomedullary junction. You see those are blood vessels, very prominent. This is the renal papilla, which enters into a minor calyx, but remember this one is a kidney that has only one pyramid. So that will be a calyx come the pelvis. A higher magnification of the pyramid displays the presence of several tubules, as you can see, and maybe a high magnification will show you that the tubules are either lined by simple cuboidal epithelium, as we can see for that particular large tube. Maybe these are collecting ducts. The other tubes which are smaller, they could be the thick segments of the loop of Henle, like those ones, or they could be thin segments. The thin segments in particular will be lined by simple squamous epithelium, like in that zone there, or in this particular zone there, or maybe in this particular zone, that is a simple squamous epithelium lining a tubule, so that a thin segment of the loop. It might be hard for you to distinguish between the collecting ducts and the thick segments of the loop, other than the diameter of the lumen. Moving down towards the renal papilla, the collecting ducts usually meet to form the papillary ducts. And so these are papillary ducts generally. We can see collecting ducts join to form that papillary duct. So that's a papillary duct. In this particular one, that's most likely a collecting duct. And look, that is a thin segment of the loop. At the renal papilla, we can see the openings of the papillary ducts into the renal papilla. Remember, these multiple openings constitute the area cribrosa of the renal papilla. The summit of the papilla is the area cribrosa, where we have multiple openings of the papillary ducts. They empty into the minor calyx. Now, the minor calyx will be lined by a unique type of epithelium, which we call the transitional epithelium. But I want us to talk about transitional epithelium in lecture 4 of the urinary system, where we're going to talk about the urinary passages. So for now, just understand that the epithelium that line the calyceal system is transitional epithelium, or what you call urothelium, but its structure we'll discuss in the next one. Good, so that is it for the histology of the kidney. Much of the information that I want you to take home is largely within the renal parenchyma. Thank you very much. So that will be the end of this particular session, this second session that focused on the structure of the nephron and histological structure of the kidney itself. The third section will focus now on the physiological mechanisms that explain how urine is formed, the three basic renal processes. Thank you very much. We'll stop there for this particular session.